Hey, ladies and gents, I am filming this in November, so if my timing is right, though, you're probably not seeing this until after Christmas break, maybe. So, which means you've probably forgot things, let's face it. All right, so you're gonna wanna go back and double check some old DNA stuff, but I'll give you a quick rundown. DNA, made out of nucleotides. DNA is a nucleic acid, polymer, macromolecule, remember that, right? And the monomers that make up that polymer, nucleotides. What's a nucleotide made out of? Phosphate, sugar, base, right? That's one nucleotide. Nucleotides line up, and then another set of nucleotides line up, and they're attached to each other, and that's your DNA. The bases are the code. They're letters of an alphabet. What order they are in, just like letters of the alphabet, what letter they are in says something. Today we are going into RNA. What was DNA? Deoxyribonucleic acid. RNA is ribonucleic acid, but it's also made out of nucleotides. Phosphate, sugar, base. It's a different sugar, a ribose sugar. But it's going to behave differently. We are going to use RNA to make your proteins. We are going to handle these notes a little differently than usual because this is the chapter that usually confuses people. So we are going to do this twice today. Yeah, I can hear you complaining. Twice. All right, I'm going to do a quick rundown, big picture of everything all at once over here on my whiteboard. And then we are going to jump into the actual notes and look at the details. I will try to make it not as long, but no promises. So let's jump on in. Today we are talking about how to make a protein. What's that? What's a protein? For shame. Rewind. Back to chapter 2. 4 macromolecules, carbohydrates, proteins, nucleic acids, and lipids, right? Well, proteins and nucleic acids are going to work together. Those are the 2 macromolecules we care about right now. This is about how to make a protein. Why is that important? Because that's what you're made out of. All of this is made out of proteins. So you got to build your proteins to build your body. So where does that come from? Right here in the cell. That's right, this cell that I drew back in homeostasis? Wow, it's a long time ago. All right, so zoom in to right here, ladies and gentlemen. This is the nucleus of the cell. What's kept in that nucleus? That's right, DNA. What? Did somebody remember something? Not just DNA? That's right, the nucleolus. Let's take a look really quick. Here, this nucleus. All right, I blew it up bigger over here. That's your nucleus. There's all these little holes in there. Those are your nuclear pores. Holes in the nucleus so things can come and go, right? I'm going to erase a chunk of this. Why? Because we want to see the inside of this nucleus. Camera angle. There. Nope. There's camera angle. Okay, cool. So cut a hole in your nucleus. All right, there's the hole in the nucleus. And inside that nucleus, ladies and gentlemen, is another structure that is not, marker's dead, is another structure called the nucleolus. All right, yes, you have DNA floating around in that nucleus. The nucleolus is going to make RNA. RNA nucleotides, not the full RNA, just the nucleotides, right? These nucleotides are going to come out of the nucleolus. Camera. Those nucleotides, RNA nucleotides, are going to meet up with the DNA and form a chain of RNA. How's that happen? Let's take a look over here. If you remember the last set of notes, we talked about DNA replication, how to make brand new DNA, right? Remember, DNA is also made of nucleotides, just like RNA we just talked about. And this DNA splits in half, and then brand new DNA nucleotides start filling in the gaps, and then we get 2 strands of DNA. Good recap, everyone. How do you make RNA? The same way. All right, the DNA is going to split apart, but this time instead of DNA nucleotides, we are going to get RNA nucleotides coming in to fill in the gaps. And notice it's a different color because it's a different molecule. We use the DNA to make RNA. And now that we have the RNA, it's going to get off and float away. And now that the RNA is gone, the DNA is going to come back together and be saved for later so we can use it again later. Let's talk about that RNA then. Here's that nucleus. Remember how the nucleus has the nuclear pores, the holes in it so things can come and go. Those things are the RNA. And this RNA is going to snake out of the nuclear pore. By the way, that's how I draw RNA. Because remember, it was only one strand instead of two strands. We made one strand of RNA, and that's floating out. And then look around. Let's zoom. Oh, wait. Zoom. See those little dots floating around? What were they again? Ribosomes. Let's zoom in on a ribosome. Right there. There's your ribosome. It's actually two pieces that come together. Yeah, ribosomes are made of two pieces, not one. And in between these two pieces, that's where that RNA is going to go. It's going to come in, feed into it like a dollar going into a vending machine. And then, remember the bases? I'm drawing this very simply. This should be phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then on each sugar is a base. And this is just going to move on in here. Now, what's that doing? The ribosome is reading this RNA. By the way, do you know the ribosome is also an RNA? Yeah, it is rRNA. That little R is for ribosome. Ribosomal RNA. And this is mRNA. That M stands for messenger. This carries a message from the DNA. That DNA has information. This RNA has the information. And then there is going to be a third type of RNA we call transfer RNA. What does it mean to transfer? It means to move something. So this is going to be moving pieces of a protein. Hey, what are proteins made out of? What's that chapter 2 bit of information I'm expecting you to remember right now? Proteins are polymer. They're made of monomers. What's the monomer? Amino acids. That's right. Good memory. So this tRNA that I'm going to draw very poorly. tRNA, by the way, this has bases too. 1, 2, 3 bases. That's important. Remember, 3 bases, and then you got all these bases here. Remember base pairing? We'll get back to that. This thing is carrying one of the amino acids along for the ride, and it is going to go into the ribosome. And then 1, 2, 3 bases. 1, 2, 3 bases of the tRNA with the amino acid. They base pair. A's with U's, C's with G's, G's with C's. That's how they know where to go. They use the base pairing to know where to put this amino acid. And then another one shows up. And this one is going to pop off and get attached to that one. And this guy's just going to float away. And then this dude's going to come in with his amino acid. And then these two are going to pop off and attach to that one. And this dude's going to float away. And then the ribosome is going to keep moving down the line and keep adding more tRNA, and adding more amino acids until we run out of mRNA. When we run out of mRNA, the chain is complete. And we just reshape that chain to make a protein. And then that protein is going to be some part of your body. There, guys. Let's go into the notes for more detail. All right, so since we just went through the whole big picture on the whiteboard together, I'm going to try to speed through these notes as fast as I can, so feel free to pause to write down as much as you need to. So this is all about RNA and protein synthesis. First off, the structure. Your RNA is a nucleic acid. Therefore, there you go, is a nucleic acid. Therefore, it is also, just like DNA, made out of nucleotides, which is a phosphate, a sugar, and a base. The difference is it's a different sugar. DNA has a deoxyribose sugar. That's where the D comes from. RNA has a ribose sugar. But yes, your 3 parts, the phosphate, sugar, base, and it's going to behave just like the DNA did, with the phosphate hooking to a sugar, and this sugar hooks to another phosphate, then another sugar, another phosphate, another sugar, all the way down. And the bases are going to behave in a very similar but slightly different way. RNA has a single helix shape, looks like DNA, but split right down the middle. The vertical part, the blue part, is the phosphates and the sugars, and the horizontal part, the colorful parts, are those nitrogenous bases. So take a look at it. This is the DNA, right? Phosphate, sugar, base, and we got the 4 bases, A's, T's, C's, and G's. You remember that, right? All right, well, here it's going to be a little bit of a difference. Ready? Check it out. Boom, different. Yeah, not that different though, right? Yes, I made the sugar a different color because it's a different sugar. Wait a minute. You guys notice the other difference here? That's right. DNA has A's, T's, C's, and G's. RNA does not have the T. A, U, C, and G. That U is different, but don't worry, guys. It's going to act just like a T. All right. Why does there a U instead of a T? I didn't make that decision. That's just how it is in nature. But it's going to behave exactly the same. Where A's paired with T's, A's can now pair with the U's. Just don't let it confuse you. So RNA does not have thymine. Instead, it has uracil. That's what the U stands for. Remember, A is adenine, the T was thymine, C is cytosine, G is guanine, and the U is uracil. They still pair up. And when they pair up, when they link together, the phosphate sugars, phosphate sugars, phosphate sugars, that is what we call a strand of RNA. And just like I showed you, the bases sticking out. This particular one is a type we call messenger RNA, or mRNA. Right? That's the one I showed you on the whiteboard. I showed you all of them on the whiteboard. This is the one that I showed you being made. This carries the message from the DNA. The DNA says, this is how you make a protein. The RNA is like, gotcha, boss. I'm going to write that down. And it is going to carry that message in groups of 3 we call codons. I believe that's a vocab word. Codons. 3 bases in a row that code for an amino acid. Right? I'm not going to expect you to memorize all these things, by the way. There are a lot of codes. I am going to need you to know them, but I'm going to give you a handy dandy chart to help you figure that out. This chart to be exact. So 3 bases. We'll say AUG was the first 3 letters. So we start in the middle, and then we work our way out to the amino acids. So it was AUG, so A, and then U, and then G, methionine. That's the name of an amino acid. That's your first amino acid. And then your second codon was AAA. So start in the middle, A, lysine is your second amino acid. And you're going to keep doing that until you get all of your amino acids in the correct order based on the order of the mRNA, which was based on the order of the DNA. You see how it all connects? DNA says a thing, so mRNA says a thing, so the amino acids line up, and you make a protein. How do they get there, though? How are the amino acids going to show up with another type of RNA called tRNA? tRNA, this is your 2-dimensional picture, this is your 3-dimensional picture, and here's the picture that most textbooks use because they are lazy. What's important is it carries 1 amino acid on top, not drawn, but it is here, and it has an anticodon. mRNA has a codon, so what pairs with it? The anticodon. 3 bases here that pair up with the 3 bases here. So if that's at AAA, this will be UUU. And once they get together, something actually needs to be doing the work. Something needs to be assembling. And what did we learn in chapter 1? What assembles a protein? Ribosomes. And we are now calling rRNA. Ribosomes are a type of RNA. Ribosomal RNA. That's what the little R stands for. It's going to be used to hold mRNA and tRNA together. They collect all the amino acids from the tRNA. So the mRNA comes in like we saw on my whiteboard, tRNA shows up, and then the rRNA, the ribosome, connects all of the amino acids to make a protein, as shown in this link. All right, so this video is a little bit blurry, but I think it should be okay. I'm going to do a quick fast forward.
So just like in DNA replication, we saw helicase unwinding and splitting the DNA open, but instead of DNA nucleotides coming in the base pair, we were getting RNA nucleotides to come in base pair. And another protein called polymerase, just like it did before, attaches them. And when the whole thing is assembled, instead of sticking around like DNA does, the RNA is going to lift off of the DNA. Any second now. Come along. There we go. The RNA lifts off and floats away, and then the DNA will come back together. Now this RNA is going to leave the nucleus, go out into the cell, where all the ribosomes are floating around waiting for them. And there we go, one piece of ribosome. They're going to match up. Remember, this has the code, and this piece of RNA, ribosome, is holding this code in place. And now our next type of RNA, the tRNA, is coming in off on the side, and you can see it has 3 bases down here. This is the anticodon pairing up with the codon. Codon, anticodon. That's how it knows where to go. And then there's your second half of the ribosome. Don't know why that came in late. And now they're all together, and down here, this little green square is the amino acid. Another tRNA comes in, base pairs. And the ribosome is going to take this amino acid and connect it to the other amino acid. And that first tRNA is now just going to go float away because it needs to go find another amino acid. This guy is now holding both of them. The ribosome moves down the line, making room for another tRNA to come in with another amino acid, and where anticodon pairs with the codon, so it knows where to go. And then these two attach to the new one. And that's going to keep happening over and over and over again, making the chain of amino acids bigger and bigger and bigger until we run out of messenger RNA. When messenger RNA is done, our protein chain is done. All we have to do now is bend that chain into the correct shape for it to do its job. It is now a protein.
So that whole thing we just saw, we saw it on the whiteboard, and then we saw it in a video, so let's just have it to put in the notes. I'm going to go quickly, so feel free to pause and write the information down as needed. Transcription is what we call the first part of this. The second part is translation. Transcription is what happens in the nucleus, making RNA. Starts with the DNA. This side should be upside down. I made a mistake. I am sorry. But the DNA splits in half. And then RNA nucleotides base pair. This has a T, so an A came in. And then a C comes in with a G. And new pieces keep, the U with an A, G with a C, new pieces keep adding in until this side looks just like this side. A and A, C and a C, T, oh, okay, U, but basically the same thing there. G with a G, A with an A, T with another U, C with a C. Almost identical. Same message. And then that ribosome flies away. The DNA comes back together to be used in a later day. That is transcription. But here's the problem, guys. That is written in a certain language. That is written in the language of nucleotides, language of nucleic acids. Remember the macromolecule nucleic acids? We are not trying to make nucleic acids now. We're trying to make a protein. That's a different language. So we have to translate that language from the language of nucleic acids to the language of proteins. So this whole process now is called translation. Just like going from English to Spanish or Spanish to English. We're going from nucleic acid to protein. The mRNA that we just made moves into the ribosome like we saw in the video. The ribosome reads the mRNA and knows which tRNA goes in that place. Lets them in. By the way, we start with something called a start codon. AUG is our start codon. If you remember our codon chart, AUG was methionine. That starts every single protein. Every protein in your body starts with methionine. So every mRNA is going to start with AUG, and that's how you know where to begin. The rRNA, the ribosome, finds the tRNA, puts it in the right spot because the anticodons pair up with the codons. And if the tRNA is in the right spot, then the amino acid's in the right spot. So remember that, guys. This is the important connection. Remember, guys, this is the important thing. DNA has bases in a certain order. mRNA has them in the same order. So the tRNAs line up in that order so that the amino acids are in that order. It's all about getting the order that DNA had into the amino acids order. That's what we mean by translation. Just like going from Spanish to English, we went from DNA to protein. And that's going to keep happening over and over until you're done. And you reach what we call a stop codon. We have 3 of them, by the way: UAA, UAG, and UGA. UAA, stop. UAG, stop. UGA, stop. They all just tell you you are done. It's like the period at the end of a sentence. When you are done, you have what we call a polypeptide or polypeptide chain, a chain of amino acids. And like I said before, all you have to do is bend them into the correct position. And then you have a protein ready to do its job. Polypeptide chain bent in the right shape is a protein. All right, guys, so that is it for the notes today, but we're not done. We still going to have another set of notes on mutations, which is a mistake somewhere along the line in making that protein. We will talk about that later. Right now, go back to Google Classroom and see if I left you any work to do. Until then, I will see you in class.